this lecture, we are going to talk about part 1 of translation, and we focus on ribosomes. Main points of the lesson are prokaryotic ribosomes, eukaryotic ribosomes, and 4 binding sites of ribosomes. The synthesis of any biopolymer can be broken down into 3 steps, that is initiation, elongation, and termination. We have seen this division in case of DNA replication as well as for transcription. In case of translation, that is protein synthesis from messenger RNA, all of these 3 steps, that is initiation, elongation, and termination, takes place on the ribosome. The process of translation is similar in prokaryotes and eukaryotes. So, from simplest bacteria to ourselves use the same basic mechanism of reading and expressing genes. So, fundamental to life as we know it, that it is referred as central dogma. So, information passes from genes, that is DNA, to an RNA copy of the gene, and then RNA copy, it directs the synthesis of protein, that is polypeptide chain. The first step of central dogma is the transfer of information from DNA to RNA, which occurs when mRNA copy of the gene is produced. And the second step of central dogma is the transfer of information from mRNA to protein. The 2 steps of central dogma are taken together as a concise summary of events involved in the expression of an active gene. So, biologists refer to this process as gene expression. A ribosome is a complex macromolecule that is composed of structural ribosomal RNA and catalytic ribosomal RNA with polypeptide chain. In eukaryotes, the synthesis and assembly of ribosomal RNA that takes place inside the nucleus in nucleolus. Nucleolus, that is known as factory of ribosomes. In prokaryotes, ribosomes, they exist in cytoplasm, but in eukaryotes, ribosomes, they exist in cytoplasm in free state, as well as they are also attached to endoplasmic reticulum in attached form. Mitochondria and chloroplast also have their own ribosomes, and these ribosomes look more similar to prokaryotic ribosomes than cytoplasmic ribosomes. So, in eukaryotic cell, ribosomes which are present here in cytoplasm, these ribosomes are called eukaryotic ribosomes or mammalian ribosomes. And in eukaryotic cell, mitochondria and chloroplast are cytoplasmic organelles present in the cytoplasm. They contain their own ribosomes, and these ribosomes which are present inside the chloroplast and inside the mitochondria, they are called bacterial ribosomes or prokaryotic ribosomes. Ribosome has 2 subunits, that is small subunit and large subunit. So, ribosomes dissociate into large subunit and small subunit when they are not synthesizing proteins and reassociate during the initiation of translation. Ribosomes are molecular machines that translate genome into protein. This is prokaryotic ribosome and this is eukaryotic ribosome. Ribosome consists of 2 subunits, one is small subunit and second is large subunit. Prokaryotes, they have 30 as small subunit and 50 as large subunit for a total of 70 as when assembled. What is S? S is Swedberg unit, not additive value. Mammalian ribosomes, they have small 40 S subunit and large 60 S subunit for a total of 80 S. So, the small subunit is responsible for the binding the messenger RNA template, whereas the large subunit sequentially binds transfer RNAs. In bacteria, archaea and eukaryotes, the intact ribosome has 4 binding sites. One binding site of the ribosome holds the messenger RNA template strand. As we can see, this is messenger RNA molecule which is directed from 5 prime to 3 prime, and the small ribosomal subunit that binds with mRNA template strand, and rest of 3 binding sites accommodate tRNA molecule at A site, P site and E site. The P site is called peptidyl site that binds the tRNA with first amino acid, and the A site is called amino acyl site that binds the tRNA carrying the next amino acid to be added in growing polypeptide chain. The E site is called exit site that binds the tRNA without an amino acid because that amino acid is already added to the polypeptide chain. So, tRNA moves through these sites successively during the process of elongation. Relative to messenger RNA, the sites are arranged from 5 prime to 3 prime in the order of E, P and A. The incoming charged tRNA that enter the ribosome at the A site transit through the P site and then leave via the E site. For the ribosome to function, it must be able to bind at least 2 charged tRNA at P site and A site, so that a peptide bond is formed between the amino acid. The ribosomes have double function, that is decoding function and enzymatic function. Decoding function means a long chain of amino acid, that is a polypeptide chain synthesized on the surface of ribosome as it decodes the information from mRNA molecule. The enzymatic function of ribosome is the formation of peptide bonds between the amino acid. For the formation of these peptide bonds, it requires an enzyme, and that enzyme is called peptidyl transferase, and this enzyme resides in the large subunit. Now, each mRNA molecule is simultaneously translated by many ribosomes, all reading the information from 5 prime to 3 prime. So, the complete mRNA molecule with many ribosomes, that is called polysome. Let me briefly restate the main points. The process of translation is similar in prokaryotes and eukaryotes. A ribosome is a complex macromolecule that is composed of structural and catalytic ribosomal RNA and distinct polypeptides. Mitochondria and chloroplast, they have their own ribosomes, and these ribosomes look more similar to prokaryotic ribosomes than cytoplasmic or mammalian ribosomes. So, assembled prokaryotic ribosome is 70S and assembled eukaryotic ribosome is 80S. The ribosome has 4 different binding sites for RNAs. The small subunit is responsible for binding the mRNA template strand, and the large subunit has rest of 3 binding sites at A site, P site and E site for tRNA.